Hello everyone, I'm Asfakur Rahman. The poster title that I'm presenting is Novel Strategies to Enhance the Efficacy of Sanitizer in Mitigating the Risk of Foodborne Pathogens. We use three strains of Listeria and three commonly used sanitizers, and they were subjected to 8 concentration and 4 treatment time to test the susceptibility, and the survived and the parent strains was then subjected to assay the biofilm forming ability and their pathogenicity. Listeria forms listeriosis. There are some current outbreaks which includes some fresh produce or even frozen foods. It is for this reason this organism is very resilient to tackle. Many food industry practices GMP or GHP to tackle this, but it is very tough to control this. In this study, the main objective is to in vitro assessment of susceptibility testing and the resistance development of Listeria species when subjected to different sanitizer, and to determine the sublethal exposure of sanitizer on the biofilm formation ability or pathogenicity of Listeria. Methodology, we use these three strains and these three sanitizer at 4 treatment time and 8 concentration. First, all the sanitizers was activated and all the strains were activated on, and all the sanitizers were prepared. The sanitizer was tested for pH and their concentration, and after that they were subjected to testing at 96 well plate. After that it was neutralized by neutralizing broth, and then they was plated in TSA. After 24 hours it was counted for the colony reduction. Repeated exposure testing, all the strains were reactivated in TSBY, and the same procedure as first time was applied for the survival testing of previously exposed strain. Multiple testing, exposure testing was done in Muller-Hinton broth containing very lower concentration of sanitizer, and 24-hour incubation. If there was any viable cell, then it was transferred to consecutively higher concentration, and if there was no viable cell, then it was transferred to fresh Muller-Hinton broth, and the highest concentration where the cell can survive was determined. For micro tier biofilm formation assay, all the strains were activated and they were grown in TSBY for 24 hour, 48 hour and 72 hour, and after washing they was the biofilm was measured in absorbance at 590 nanometer. Adhesion, invasion and intracellular growth was carried out by cacocell and bacteria incubation and followed by flow cytometry for the pathogenicity testing of this Listeria strains. In susceptibility testing, we can see that from 32 to 4 ppm there was no significant reduction by all three sanitizers, but from 512 to 64 ppm there was a significant reduction of the bacterial population. And in repeated exposure we did not observe any significant difference in the behavior of parent and previously exposed bacterial strains. For multiple tests, the result we found was at 32 ppm of benzyl choline chloride and 64 ppm of uh, uh, peroxyacetic acid there was the highest where cells can survive. And for biofilm production assay, we found that there was no significant difference in parent or previously exposed strains in case of biofilm forming ability. Adhesion invasion test reveals that the pathogenicity there was no difference in parent or previously exposed strain. Future we will do the ACM for this test, and this study acknowledged the University of Texas Rio Grande Valley Startup Funds and Dr. Neera Karsavu. Thank you very much.